Thank you very much. So also thank you very much to stay with me right now. And so, we have heard a lot, great, presentation this morning and a different molecule, different modalities, and also hopefully you can hear me. So also, have a great, in not only the in vitro data, but also a lot, in vivo data. So for me, as a ProBio, so I like to talking about, how we can work with you like a partnership, and so you spend so much energy, time, and also funding, to develop these great molecules. So as a CDMO company, ProBio, how we can work together, to bring your excellent molecule into the clinical trial IND filing and win the market, win the speed. Speed is also very important. So we like to work with you to develop a great molecule together and bring your market into the market. So I was talking about a little bit about our company. So move forward, right? Okay, maybe this way. Okay. Okay. So first off, I would talk a little bit about our company. And the ProBio actually was spun off about 3 years ago from GenScript. And right now we are legally, financially independent company. Our headquarters are located in New Jersey. And so we have that, and we have a very good finance funding. And early this year, we got like a $220 million. So we have a capability to expand our services, globally, and hopefully, we can do a great job, to support our industry to bring the molecule into the market. And you can see here we have excellent R&D facility. So we have a lot, activities inside to even, like I heard, develop great molecules, specific molecules. So we also can help you work together to develop world-class, therapeutic molecules. And also, we have the facility, manufacturing facilities, and not only GMP, QC, but also we have a fill and a finish line. And so we do everything within the same organization, and so can minimize the risk and also cost effectively. So hopefully, we can, that's the manufacturing here. We have a kind of like a small scale right now, but we also build up the large scale. So we'll be ready early next year. So we really can do the one-stop solution or service, within the same CDMO company to really, to do the fast pace, cost effectively, and bring your product into the clinical trial as quickly as possible. And so far, we have a very clear track record, and you can see for discovery part, actually we have done almost 1,000, leads to help our customer. All this, for the different modality, antibody, bispecific, tri-specific, all this kind of different molecule. And also we have 13 therapeutic antibody license deal right now. And for the CMC part, we have developed about 200 commercial cell lines to support the R&D filing, and also about 90 CMC and a CMO project. And actually here we already, got the 26 global R&D approval and also deliver more than 100. Oh, I'm sorry.
Oh, no. Real quick. Make sure you're looking at the screen on that one, because then our virtual audience only sees like the side.
So look at here.
Yeah.
Oh, okay. I tried to point to the. Okay, maybe I cannot use the laser point. So okay, so we deliver more than 110 GMP batches. Okay, and also we have done a lot audit and globally, and also passed all the regulatory inspection. And this one show you some just a snapshot about our project for different companies and different projects. You can see different modalities, and also they're in the clinical trial in the USA and also in Asia. So now I try to touch a little bit about our drug discovery department or platform, because I hear this morning a lot of great molecule designing. So what maybe if you need help, I trust you have a fantastic R&D team, but maybe we can work together. Maybe we can support something for your molecule development. So here we have a different platform, hybridoma phage display, single B cell, and a transgenic mouse to develop the different antibodies, monoclonal, single domain, and also bispecific we're talking about today. And we not only we help you generate leads, but also we can do humanization about the 3 weeks very fast, and also affinity maturation studies about 8 weeks to really enhance your capability. We really do. Okay, so if you need work with us, so we can further improve, just like a previous talking about the specificity and also affinity. So that's we can work together also if you need it. And also we can test the functional assay, make sure the molecule we develop still maintain good quality and also efficacy. So we have the system to test all the molecule when we work together. And here just some bispecific modality to show you the capability that we work with our partners. It's not the titers and also the structure stability, and the way that can be purified, the purity and also recovery, and also whether can maintain the functional activities. So for example, we done different modalities for the customer, but also our in-house, we have another format. I think Dr. Lu also mentioned this morning, they do similar, this kind of project in this morning, and find IgG one is great, efficacy compared to other IgG formats. So that's a little bit discovery. So I can't, because time, we limit, so I cannot touch that. But I like to focus on more time, more on the CMC, services, what we can do, because at the end you want to get into the clinical trial, and you want to make sure the purity, stability, efficacy, right? Everything, make sure the molecule after manufacturing, the molecule really good quality, you can do the clinical trial. So we minimize the risk. So that's the key. So what we can do, and we have the in-house cell line and the expression vector. So for example, we have an antibody, and the titer by average, we can get more than 5 grams per liter. By specific antibody, usually, kind of a little bit more challenging, right? So we can, by average, we can get more than 3 grams per liter. So that's our track record right now. And also we have excellent downstream team analytical purification formulation. So imagine, a lot of molecules, they are not naturally exist like IgG. So a lot of acid, they are all processed, they are not available or not optimized. So how we can ensure the purity and also the structure, stability, everything, right? We want to make sure the molecule has a good quality. And I mentioned here already, so I don't want to mention it right now, but we can do a lot, GMP capability, flexibility also. So when we work with our customer, also different process, manufacture process, we also optimize. So here is a kind of like what kind of molecule distribution percentage we have done. So I'd like to just focus and point out a little bit, we do have about 46. They are more challenging molecule, but specific, tri-specific, also recombinant protein. And again, they are not available a lot of time for your process, downstream process. Okay, there are a lot of challenges. And the right side, there's some modality we have done to help our customer get into the clinical trial. And so, as I mentioned, and because there are a lot of molecules, maybe they are not naturally existing molecules. So you do see some challenges like titer, low titer, mismatch, byproduct, a lot of byproducts. So how you can solve the problem or minimize the problem from the beginning, and also how you can develop and optimize your process downstream. Each step, they are critical, right? To ensure at the final, steps, so you will have a very good, purified protein or molecule to support the R&D filing. So I mentioned here already, and this we have the in-house CHO cell line, but I like to emphasize a little bit this one. Maybe you don't need it, but maybe you need it. But this one is we call CHO K1 ADCC, which means the fucosylation, the genes was knocked out, so can enhance if that is the ADCC dependent, so we give you another option. So just keep in mind if you need this one. So we do have it. We have the old license in the IP, and so you don't need to worry about that. In the meantime, we have the high expression vectors. So when we have this all combination systems, as I mentioned, we do have a very good productivities, right? And also for stable cell line generation, we have the whole high throughput systems. We have a VIPS, this seeding technology to ensure one cell per wear, to get more analysis. And also we have a cell imaging system to really capture the image to ensure we have a single cell, so meet the regulatory criteria. And after we identify and screen out the top cell lines, we also have an EMBER system to validate your top candidate, and also to do some DOE studies to optimize the culture conditions, right? So really make sure that productivities, efficacy, quality, everything, from the beginning. So this one gave the case study. So this is a asymmetric bispecific antibody. So we didn't find if you use a single vector, actually you will see a lot of byproducts. However, if we do co-transfection, then you see very good, the 72%, that's your main products, right? Versus here, not only the low titer, but you can see very, very hard for the downstream to get a good purification and a good recovery. And if you see right side here, and the titer increase tenfold, more than tenfold. So really can do some optimization from the beginning to minimize your issues. So if you, during the process, develop a process, we can work with you, to different testing. And this is another case study. So in terms of how to improve titer, maybe in the audience, you already have a lot of experience, but for the research, the early side, maybe not. But for the process development group, they do have a lot of experience. For example, how you analyze the spanner meeting to look at the nutrients, what kind of new amino acid was depleted, so we can analyze that. Then we can do the supplementation. And the CHO cell line to CHO cell line, actually, we all call, for example, all call CHO K1, but actually CHO K1 to CHO K1, they are different, and they may have a different sensitivity to the pH. So we can do some testing, and also how to improve the titer. If you change the seeding cell density from the beginning to a high cell density, like a 5 million, 10 million cells per ml, then how to improve the titer. But if you do that, you also have to optimize your feeding strategy because you change the production process, right? So you have to optimize. And here, temperature is a common, but you do see molecule to molecule, they are different. So maybe C, maybe not. But you also keep in your mind, we can do all this kind of process development to improve your titer. And quality-wise, so glycosylation, silylation, so we do have the experience to use different additives test different concentrations and a different time, and to improve the glycosylation, from 49, 42% to up to 80% like that. And also you can test different base medium from the beginning to really screen out which one will be the best to meet your quality spec. So that's the upstream. What about the downstream? So my presentation, I try to give the whole picture from early, then the downstream purification analytical. So you see our capability, how we do the solve the problem. So that's the reason I gave you different case studies. So for purification, you may face the aggregation, degradation, or mismatch, so all from different byproducts. How you to remove them, right? And so you can use different chromatography. But look at each single chromatography may be not big a deal, but the good thing is how you can combine together, how you develop the whole process, work together to remove the impurity, but also get a good recovery. You cannot say my recovery only 20, 30%. Right? You lost a lot of products. So that's, you need to do, we call it PD work, right? And also combine with the elution buffer, the salt concentration, different, what kind of salt, what kind of pH value. So that's the whole process. You put it all together to get a good purity. So here is the case study. And you can see initially very low recovery to remove aggregates. Then you use a different column chromatography, then you can improve the recovery. And also the purity from 96 now even up to 99%. So further improvement. And for homodimer, this is a byproduct. And you can use, and initially use affinity column, you will see it. 39, right? So about 39 impurity. Then you use a different column, then really bring down the impurity and improve the purity. Okay, so just give you some idea, about our process, our capability, how we can work together. And also during the process, you may use different pH. And so, for example, we try to improve the aggregation issue or solve this issue using different pH conditions. But also you need to be careful about the degradation, the stability, and the different environment or conditions. So that's a purification. What about the analytical? So analytical also very important to make sure you have a right assay to distinguish your products from the impurity, or you have to measure the test the functional assay, MOA, and how about, other impurity, do you have the right method or validated method you can trust, during the whole process, right? And also, actually, I mentioned the cell-based assay. Actually, we have excellent cell line engineering team and to develop all these cell-based and to support your molecule development and test the function activity. And all this assay, by the way, is we have a clear traceability background to support the IND filing. Okay, and also we have a great quality systems. So this gives you just a couple of maybe example for the analytical department, their work. And for this molecule, they probably have a similar molecule with, but they do have a different PI. So you can develop ICIEF method to distinguish the products from your byproducts, okay? And for right side, if they have a similar PI, but maybe they have a different molecule, so you can develop a CSDS method to distinguish or monitor the whole process. Right? So what about the function activity? And you want to make sure the molecule you develop by the end of, after purification, everything, they have a maintained function activities, right? So for this one, target TNF alpha, interleukin 17A, and we use one cell assay HT29, and also you can see complete block, this two molecule, you have to add these two molecules, then block this bispecific antibody functional activities. And this is a tri-specific antibody, T-cell engaged, anti-CD3, and then anti-2 tumor antigens. Antigen 1, 2, and 2 independent cell-based assay. You can see very nice function activity. So we always check and use available, or we develop the cell-based assay to really ensure after the whole process, your molecule maintain good functional activities. And again, this is just a few examples. We have an in-house, available cell. They have all clear background traceability to support the IND filing. Okay, so it's available. We do the testing after we do the whole process from stable cell line generation, purification, formulation. Okay, so but by the way, if they are not, you don't have your molecule is brand new. Actually, we can work with you together to develop the cell-based assay. So this is just summary for my current information or talk. So we have we know how to improve your titer. We know how to do the purification and analyze your molecule, make sure your molecule have a good function activity, high quality, efficacy, and so we can support all the documents to support the IND filing. So finally, I'd like to touch a little bit about our quality system, because this is absolutely important. So we have a great quality QA, QC teams, and over 70 people support all the projects around the world. And from beginning, the raw material control, environment, and the safety, and the whole process, and also lab, raw data capture, electronic data, and also comply, to all the regulatory criteria, and FDA, US FDA, EU, and also Asia Pacific. And so far, we have done a lot, audit, and there's no problem at all. So that's our track record. So I hope we can do be partner with you and work together and make sure we can help you if you need a CDMO company services. So we can work with you to ensure you can get into clinical as soon as possible. Thank you very much and for your time. And questions? Yeah. Any questions?
Any questions?
By the way, stop by our booth if you have.
Questions for our presenter? All right, give him one last round of applause.